Thanks everyone for coming. This is great. It's a little bit different from the dinosaurs, but not so much. Dinosaurs have DNA. In fact, almost everything in this room, in this building, has DNA on it or in it. And someone's going to say, well, what about the mineral room? Someone touched it. There's DNA floating in the air. And we'll get into a little bit of that. We'll pass it through. What I'd like to go through tonight, it may be in a few minutes since you've had DNA and RNA, that high school biology. So we're going to do a little bit of that, but I want you to have fun. And there's going to be a few slides that might ask some questions that make you a little uncomfortable. That is part of the purpose. I've been coming to the Delaware County Science Institute since I was about 6. So that's more than 50 years. The chairs are identical. The comfort level, minus the cushions, are identical. So let's have some fun. And so this slide sort of encapsulates some of the rapid changes that we're experiencing with genomics. The sequencing is new because the technology of sequencing is different. It has changed appreciably. A lot of what was there didn't exist in the early 80s when I was getting my doctorate. Not at all. There are massive computers that analyze. So we talk about sequence, and then we analyze the DNA, and we learn about the DNA and the RNA and all the other things. So these are massive cloud computing. Back in the day of saying, I will take my desktop laptop and I will use it unless you have 500 gig of RAM in your machine and willing to go for 3 or 4 months, you probably will not be able to do that. But now it's in the cloud. No one needs to buy any of that stuff. So we're learning very, very quickly. It's very exciting, and I want to share some of that excitement with you. So with this, we'll have a little bit of a refresher. I know it's that a hot minute since you had DNA and RNA in biology, and there might be a few of you who are genetic counselors, so just bear with me. It will be painless. I went a little too fast. Okay. Now we are caught up. Okay, and then, so technology is driving the change. That's why we've gotten where we have, as well as there's a lot more researchers studying it. It's starting to be involved in a lot of other fields. We're going to talk about human biology. We could be talking about plant biology, or bacterial biology, or viruses. We'll talk a little bit about bacteria and viruses as they relate to human biology. We won't really talk about plants. We're going to save that for another time. And there's something rather strange. The more that we learn, the more we realize we don't really understand what's going on. Back in the 80s, we called it junk DNA. That's a cop-out to say, I have no idea what it does. Well, now we're starting to get a clue of what it does and how it impacts our lives. So we're going to talk about what's new, where it gets used, where it touches your life every day. And then we're going to highlight some of the concerns. Because hopefully it'll get very exciting, and this is fun, but there is a dark side. So the old dictum, DNA is your genes, that's where everything is encoded. It gets transcribed into RNA that goes out and, after a fashion, makes proteins, and sugars go on those proteins, and that's how you are. And that is still true, somewhat. But then, and in the 1960s, that's what we learned. Probably, if anyone's from the 1950s in biology, that was not the case, but the 60s is life is based on DNA, and then there is RNA, make proteins, and they do stuff. However, in the 80s, we had this idea that RNA does stuff on its own. In fact, 5 years ago today, our lives fundamentally changed due to RNA in a particular virus. Can anyone tell me what happened 5 years ago today? COVID. That was not a DNA virus. It was RNA. And we know the impact on many lives. Friends of ours, people who have long COVID or are not present anymore. So it had a fundamental, and that was RNA. And then in the 2000s, I learned there were 5 different types of RNA. Boy, was I wrong. Now there's 20 or 30. There used to be mRNA, messenger RNA, transfer RNA, and now they have 2 letters. I have to Google them. I honestly don't know what they all are anymore because it evolved so quickly. In the 2020s, there's all this RNA. And then there's proteins that go and tell the DNA what to do and the RNA what to do. So wait a second. I grew up with this. Simple waterfall. DNA has the information, RNA takes it, makes proteins with ribosomes, and we're real simple. So this is going to be a little unsettling. Park that in the back of your mind, and let's move into it. Or not. Come on. Okay. Oh. Okay. Where are we? Apologize. Okay, something's not running. I'm going to do that again. Yeah, actually, I'm in preview mode, which is a bit of a problem. Okay, come on. We can do without the video, but I really would like to have it. We have 2 letters in Morse code. We have 2 letters, 1 and 0 in binary. In computers, we have 4 letters in DNA. Anyone know what the 4 nucleotides are? CGT. And in RNA, it's uracil. So those are the letters. The words are 3 of those letters. TGA is a stop. So there is a sequence. We know what it is. We understand what they are and how it's done. That is your code. Your DNA, if it's unraveled, it's supercoiled, is about 6 feet long. And it is all compacted deeply into your cell. So I do have another movie, which I'm sort of wondering if it's going to play. Now with that, some words I want to share. DNA, from all the cloning movies you've watched, can become any cell in your body. It has all the instructions. However, DNA in your body takes 2 paths. One path is the germline. So this is the DNA in the eggs and in the sperm. That is what passes forward. Those are the chromosomes of having blue eyes, whether it's recessive, etc. You may remember that. That is passed on to your offspring. So if you remember Mendel's peas, etc., we are talking about the germline DNA. And then there's a same DNA, but a different path called the somatic DNA. And this is the DNA in your cells, in your organs. This is the DNA that replicates and may have variations in it, where things start to fail, where cancers may occur. Cancer, there's an idea that cancer is merely a misprogramming of the DNA, so that almost every cancer is normal stuff at the wrong time. You have cells that migrate to form the neural cord, your spinal cord, and the brain. We call that metastasis in cancer, but that's a normal function. You have organs that grow, normal function. There is a form of cancer called a teratoma, it's very, very rare. It's when the cells go nuts, and so a hair cell may be next to an eye cell, which may be next to another cell, because the programming of the DNA is off. When you're exposed to cancer agents, you tend to damage the somatic DNA. Don't tend to, unless you're being sterilized or something like that with radiation, don't tend to hit the germline. This is going to become really important later in the talk as to which form we're talking about. So the variance in somatic DNA, because of damage to your DNA, because of environmental aspects, tend to be on the somatic line. Two other words, your genotype is all about what your gene is recorded. It was written in your DNA, literally. Your phenotype is the expression of that. So I mentioned earlier blue eyes. If you had mostly brown eyes, blue is a recessive gene, doesn't tend to be expressed. It is for me, I have blue eyes. So this is the expression of your skin, your melanin content, et cetera, et cetera. That's an expression of a gene. It's turned on. Just because you may have a sequence, such as a sequence associated with breast cancer, BRCA1, BRCA1, doesn't mean it ever gets turned on. The fact it's there means it could be turned on, but it doesn't necessarily have to be turned on. What turns it on? That's a really good question. Way, way back, before Mendel, there was a sense that evolution could be in the DNA, or it could be not your genes, but your nurture. The reason giraffes were so tall is because they had to reach those high branches. This is called the Lamarckian theory. That was seen as totally ridiculous, except your environment does impact the expression of your genes. Your environment may contain viruses and DNA that become part of your somatic line, not likely the germ line. And so the environment does impact. And this goes to the cartoon all the way on the right side of the proteins may actually, your environment, nurture may impact your life as well as germline. Okay. So in the video I didn't get to see, DNA is supercoiled. It's bound by proteins called histones, which are part of the regulation, and they get bound up into nucleosomes, which are on-off switches. So just because you have a sequence, if it's turned off, it may never be expressed, which could be good or bad. If you need to have it expressed and there is a repressor gene that stops it from doing that, you don't have those proteins. if you have it turned on and it never turns off, and it decides, I'm going to grow like crazy, just like those fetal cells grow like crazy, and I'm going to shoot out all these little satellites out there, just like the neural crest cells for developing neural systems. We call that metastasis. Is it turned on and that switch is jammed on? So that becomes a target for therapy. Can we turn off things that got turned on and didn't get turned off? Can we do something about it? Oh. Okay, that went way too fast. Now let's try this one.
We'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. To the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. Based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Okay. Question. Should I go and see if I get it, or should we keep on going? We have time. Go back, she says. See if I get lucky. Okay, so physical expression. Okay. What are these guys called again? Nucleosomes, absolutely. Very good, paying attention. So hopefully I've gotten you up to speed on all the terminology. There will not be a quiz on this terminology. It's like, yeah, I remember that stuff. So what is new? Okay. We moved too far. So the sequencing, this is turning DNA, the biological specimen, perhaps someone gave you during Christmas a kit, you spit in a tube or you wave something or whatever, and you sent it off, and 6 weeks later you got something down. The sequencing is turning biology into data. And there are two main types. Sanger sequencing, very precise. The Human Genome Project started in the mid-90s. They got 80% done in the early 2000s, and they finished it in 2018. It started at $1.4 billion. And that was because they funded hundreds of labs. You'll see why in a bit. Takes a long time, very expensive, high precision. Now, there is something, and we'll get into what this is, next generation sequencing. Not quite as accurate, but you can go from biology to a sample in 4 hours, give or take. Depends on the piece of equipment that you use. And for less than $1,000 a sample. That's just the sequencing. We have data. By the way, that data, a standard input is 50, 60 gigabytes. If you've ever streamed an hour-long movie on standard definition or on high-def TV, most of our TVs, that's 2.7 gigabytes in an hour. If it's high def, you got one of those really jazzy 4K screens and you have a 4K source, that's 7 gigabytes. I'm talking 80 to 90 gigabytes just for a simple data set. Think of your Word documents. They're a couple K, maybe a meg if they have pictures in it. What's a gigabyte? Is 1,000 megabytes. 1,000 gigabytes is a terabyte. That's actually a standard set of sampling if we're measuring over time the same person. A petabyte is 1,000 terabytes. We'll get into that in a bit, but it requires a significant amount of computing capability. When we moved a biobank, the initial set for a biobank was 23 petabytes, and we had to bring it into the computer. It reminded me of the days of 300 baud modems. It took 3 weeks. And we prayed that it didn't fail in between. That's the size of the data. But when you think about it, you got it in 4 hours for less than $1,000. That's pretty good. It also has a whole new field of bioinformaticians. When I got my PhD, we went and we did the wet work. We isolated the proteins. You got your hands dirty. In fact, I do remember the RNA had contamination on my fingers because I didn't wear gloves. That's a horror story today, but that's the bioinformatics. They never get their hands wet. They sit in front of a computer. That's it. I said, you didn't isolate the DNA yourself? No, we don't do that. We just get it from a data source. So it has changed. And then there is a notion of biobanks. Think of them as libraries, where they have UK Biobank in London has 100,000 individuals over time, over the last 10 years. This is genome data, expression data. This is their RNA. This is their medical record, their weight, their history and physical, all of that. And that was 5 years a landmark. Our Future Health in another part of London said, we're going to do better. We're going to do 500,000. All of us, so these are both our UK-based. All of us came from the NIH. I think they were targeting for 1 million. And they're also disease-specific. The Veterans Association wants to do all the veterans. Or at least they did a few years ago. I don't know where they are. So what else is new? Not this clicker. Here we go. New use cases. So now you have this stuff. What are you going to do and how is this data going to change your life? The first place is in precision medicine. This is not take 3 of these and if it doesn't work, come back. I'll try something else. And that's how medicine 30 years ago was done. Now it's we have this cancer, we have this condition, we'll try this protocol and then we'll try that protocol. You may have noticed all the cancers are based on organ types. That is changing to what genes or gene regulation. And you're starting to see the BRCA1 breast cancer. You're starting to see EGF receptor positive and negative. This is not a description of the tissue. This is a description of the genome and the regulation of that genome. And so they're targeting it ideally for your cancer, for your condition, not people at large. So they're tuning it, and when you have that turnaround time of the 4 hours for less than $1,000, and they also have fairly rapid to get a plan. How do they know it's going to be good for you? We're going to look at other people that have had this therapy and how did that work for them. Direct to consumer. How many of you have submitted your saliva sample, you don't have to admit it if you don't want to Ancestry or 23andMe or someone else? Okay. It's fun. You get to find relatives. I've found cousins 3rd and 4th times removed because there were matches. They were great corroboration of what I thought I knew. I mean, the quality of the data in Ancestry is not so good, but the genetic part is much better. You may find out I'm 3% Jewish, which may have an issue with breast cancer. You may find out you have relatives that you're German or Scottish or from Oceania. It's interesting. You may find out your father is not your father. And that does occur. So there's some unintended. And the forensic science, the movie we just played, talks about identifying from the DNA, it was in no system, they identified a familial match, an uncle of someone that was a suspect. You can't use that in court, any court in the US, but it was useful pointing them in the right direction. There was use of DNA in Europe because there were crimes committed and they were sure they were committed by the immigrants coming from the Mediterranean. But then they looked at the DNA, it was from a Western European. That killed that argument real fast. So it has interesting uses and interesting consequences we'll get. Another exciting area is gene editing. So in the germline, there can be mistakes that are passed to you. This often happens in early diseases, age 1, 2, and 3, where there is a defect. Phenylketonuria, well known as a genetic disease. Others, hemophilia. I'm sure you all remember the stories of the royalty and the Russian emperor, etc., and they intermarried, etc. I have no idea about the Belgian prince who died yesterday of a genetic disease where his mitochondria, that's the energy part of the cells, just started to crap out. It is a genetic disease. It is rare, but there is now a treatment for hemophilia. There is a treatment, and I say definitive care. There's 2 types of medical care. Definitive care is to cure. You're done. Palliative care, which most of us are on if you take statins for high cholesterol, if your blood sugar is not so good, you're not making insulin, that's palliative. It is not a cure. What I am talking about now is a cure. The third one was last year, sickle cell disease. This is, it was mentioned in the movies where the red cell sickled and clotted. It is incredibly painful. And here is a disease. If you know Spark Therapeutics in Philadelphia, they had a genetic treatment for a specific rare eye disease. One of the problems with Spark Therapeutics was it's a great scientific thing, and it cost $460,000 per eyeball. And no one was going to pay for it. So the science was amazing. This is fantastic. But the commercial side just didn't work out. Others that are in the pipe that are looking good, they're in clinical trials: spinal muscular atrophy, cystic fibrosis. Now we have people that used to die when they're 10 or 12. Now they're up into the 30s and for palliative care. Imagine a definitive therapy. Okay, let's go to sequencing. The Sanger sequence is slow. It's very precise. A sequence, you pour gels, you read the gels. And by the way, they do that 1 billion times. No kidding, they divide it up on several hundred laboratories and say, you take this chromosome, you take that chromosome, and you work at it, and we'll all meet for the next 15 years. Tons of theses were written on it. Great exercise. A lot of it was really tough. Very precise, and it provided the reference genome for humans. Which one? Which human? Does it matter a whole lot? No, we're 96% the same as each of you. A little less so for a banana, but I don't really worry about bananas. The other part, which is NGS, which is sort of a misnomer, next generation sequencing, it's very fast. Matter of hours. Because you do a shotgun. You just do, I want to do the entire genome all at once. I'm going to take it, chop it into pieces, amplify it 3 orders of magnitude, and sequence it very, very quickly. This is all done inside the machine. When you send in that saliva, the last time a person handles it is opening the mailer. They put it in an assembly line, and that's it. Okay, gene sequences. When you watch the crime dramas, they swab the DNA, and amazingly, they have the suspect in the interview room that afternoon. And my wife reminds me, this is fiction. Get over it. This is single nucleotide polymorphism. And so how I like to explain that is, imagine you had a newspaper, and you could read 1 or 2 words in several of the stories, and you don't get to control where these words are. And then I'm going to tell you what was in the newspaper. Well, economy, some war, some opening of a grocery store, etc. But imagine, and this is originally the Sanger method. So you had to do an awful lot of these to get sequences together. Next, what the FBI uses is about 23 to 26 nucleotides to identify you. Next generation sequences are several thousand nucleotides per read. So imagine you had all the yellow highlighter. You have a pretty good idea of what is there. You have some missing stuff, but you have a much better idea of what's there. Now, what does it mean? That's the analysis part. And this requires huge computers. It's not unusual, and so DNA and X is in this business, the analysis, the computing. They may have algorithms that last anywhere from days to run. So the norovirus, the standard sequence is 72 hours with graphics processing units, huge machines. And they love to run them all in parallel. I've got 14,000 samples. Please don't run 14,000 pipelines all at once. You might want to run 1,000 and see how it works out before you commit. But the fact is you can't. You could be analyzing this anywhere in the world, in parts of Africa, looking at DNA that was sequenced from your people. The hard work is the analysis. The turning from the biology to the data is just a piece of equipment. Send it out. It's the analysis that's the hard part. And you may have, we'll get into this, analysis saying, I want to see everyone between 40 and 70 that's male that has these medications, et cetera, and how does it match for your cancer against those cancers on this particular protocol? How did it work out? Because it's kind of hard to have a control variable when you're the only subject. But you're not. There's storage in biobanks. I can't have a talk on computing without artificial intelligence. We're not going to go into artificial intelligence. It is playing a fabulous role. It's really early days for genomics, but what should I look for? What suppressors are out there? There's some terminology. An exon, that's 2% of the genome we saw in the second movie I showed you. That's the coding for proteins. The intron is probably gene regulation. I don't know what it is, so there's a lot that we don't know. The more we sequence, the more we analyze, the less we really know. And this is correlation. This is not causation. Because I see this, and this is what happened, I can make an inference. I didn't say because you have this sequence, you will have that. It could be turned off. Don't know. So AI, which is its own subject, so you may want to think of having an AI presentation, large language models, et cetera, in some future talk. Or not. Come on. Biobanks. I mentioned all of us. That's the NIH, the biobank. I'm involved with this one. I'm involved with this one. Not so much. I'm a researcher in this one. But the story in the top came out Friday. And one of the challenges of the biobanks I just referred to is they're incredibly Western European. And so this particular story was done out of some Australian labs, the Gabon Research, and they found that if you have this sequence, you probably have a heart condition. And as it turns out, most people in the oceanic area, it's normal. But because they're in the biobanks, it all seemed bad. And what this meant was the heterogeneity wasn't really there. You couldn't make that inference. Because when you looked at it, did you sample all the different genomes in the world? No. There's lots of stuff that is just normal. And so that's one of the challenges, is understanding what's in the sample. It's okay if it's very biased, as long as you know what it is. And realize you cannot generalize until you have a sampling in the population you want to use. This is a slide you saw a miniature of. This is completely synthetic, so don't read too much into it. Someone had a lot of fun, so they wanted to know all males between this age and that age that were on these medications. What was their diastolic? That's the lower number. I have no idea how much pizza they consumed or why they brought this up. But the idea is, can I slice and dice all of this data to say someone like you for this particular treatment? So of the 100 samples, this is at a UK biobank, 6,000 were a dead-on match. By the way, the UK biobank is now 50 petabytes. So if you've ever done a database, and it's 50 petabytes, you're slicing and dicing in real time. Those are into computer science. This is a graph database. It's the same type of technology that when you buy a baby present, Amazon reveals all the rest of the baby presents and assumes that you're going to have a baby, etc. When it's me and I'm thinking, why did I do this? That's a graph database. People like you also buy this stuff. People with these combination of sequences also tend to have this. It's retrospective. So how would they do with COVID? We would have no data prior to 2020. They weren't sampled. Not a whole lot of Chinese in those databases. And not any in the Western area. So this is a common tool. This is called a cohort browser. And then an exciting part is gene therapy. We're moving from diagnostic, because you have this, I infer you have that, to let's go fix it definitively. And so this does involve, let's move to the next slide in the interest of time, CRISPR-Cas. This is an enzyme that was made in bacteria to defend them from viruses. I did promise to talk about viruses and bacteria. So bacteria are often infected with viruses specific to that bacteria, and to take care of it, and that virus genome will go into the DNA, the somatic DNA, if you will, of the bacteria, and this is the bacteria's defense. We always have this little scissor, and in the news it's CRISPR. They almost never say Cas9. That's only a scientist would say that. It's so precise. Imagine I dumped 400 scissors into the soup, and the one that I cared about worked, and that's great. What happened to the other 399 scissors? Some of them didn't do anything. Some of them nicked some other DNA that 20 years from now we may find out was not a really good idea. But we didn't know that, and we're very happy with the results, and you get to live another 20 years. So that's why the FDA says we need to study the side effects for the rest of that person's life, because we don't really know what we did. Can't claim a victory if you kill the person 30 years from now. If you know that's the case, maybe we can treat it. So isn't that wonderful? Neat stuff. Growing. And yes, we're almost done. So what about privacy in your genomics? Okay, so if you've had a credit card stolen, it's terrible, but you can get another. That's not exactly the case if someone steals your DNA. You don't get another. There's no mulligans, do-overs, none of that. And we'll see why you care about that, or should care. So, I am sure when you've gone to the doctor's office, you have consented so the payer can look at it, maybe pay or maybe not pay, etc. You probably signed them. And how many of you, who are not lawyers, have actually read those consents? Did you read your 23andMe or Ancestry consent? Probably not. So when you give consent for genomics, you're giving it for all living family and their relatives. Did you tell your brother? Did you tell your cousin that you're doing this? Oops. Makes for interesting Thanksgiving. But here's the kicker. Oh. There you go. All of your family, how about their unborn relatives? They're kids that aren't even born. And exactly, do you have a right to give that away? I don't think so. This is a huge concern in Europe, where privacy is a right. In the US, privacy is attached to a transaction. Your credit card, that transaction. HIPAA, the healthcare, but if you're not a clinician, you can have at it. In Europe, it's your right. And genomics is a special category. And this is a huge issue. Okay. Did you know, for 23andMe, they sell this stuff. This is actually the second area for $20 million. Now, I do know someone from GSK that said, Lauren, a third of that data was cats and dogs. How could that be? Okay, so you order 6 kits, and 2 of your friends don't want to do it. And there's the cat walking by. There's the dog walking by. They swab that and send it all in. And 23andMe sells it to GSK, and they're saying, what the hell is this? GSK is no longer in the pet business. So, DNA is a natural product. It cannot be patented. However, the data and the analysis is. So when this was sold, did you get your royalties on that 20 million? If you were Taylor Swift, you'd say, I want the royalties every time my song is sung. But you didn't get anything. Read your consent. It wasn't there. Something else to think about. But there's more. You can read this title two different ways. They're monitoring for another COVID. They're having surveillance about what's going on in their country. Public health type of surveillance. Or you could look at it for saying euthanasia. Getting rid of populations. Or you could look at it as saying we want superhumans. So people have bad genes we're going to cull. This is a huge concern in Parliament. It is one of the executive orders from the previous administration that's still there in the current administration about genomic data going to China. Do I dare do it twice? So what did we learn? We had a refresher on DNA, RNA, a little cartoon that is used for public education from the American Chemical Society. Technology is what's driving the change quickly. The more we learn, the less we know. And this is exciting. This is good stuff. And this is an area we should grow. What's new? The technology, the people, the theories. We're moving very, very quickly in a state that's relevant to your care and the care of your children and your relatives, which you may or may not know you have. And then there's some areas of concern. And with that, it's 8:30.
You said, like, you can cure sickle cell and fix the genes and it no longer exists for that person. What about going forward if that person has kids? Do they pass down the gene or still?
So the question is, if someone has their red blood cells modified in the bone marrow, fix those cells, put them back in that person, and they're cured of sickle cell disease. But what about their children? The bone marrow treatment is your somatic line. What gets inherited is the germ line. So the answer is, unfortunately, no. If you were to do germline modifications, oh, those nice little scissors that seem so precise, ooh, we don't know. So it could be done, but it could be very dangerous. So not at this time. Other questions? Yes?
I have two questions. One is, what does your company do?
So DNA Nexus is a secure and compliant cloud resource. So the sequencers push the data into DNA Nexus and start running pipelines like quality, et cetera, and we host it's a multi-tenant, multi-cloud. If you're a citizen of the EU, the data must stay in the EU, so it all happens in the EU. We do that for about 65,000 customers, all the large pharmaceuticals, almost every medical device or diagnostic, some CRISPR-Cas companies, et cetera. So they use us for analysis. The regulatory agencies can look at the data. The FDA likes to look all the way down to your exact data and redo your analysis. The Europeans take the executive summary. So it is a place that's secure and compliant to do that. Our Amazon bill in other clouds is approximately 10 million per month. So we do a lot of computing. And your second question?
Second question, I'm curious what your response to this will be based on your cautions at the end. What is your position on forensic investigative genetic genealogy?
So when the green, and I'm blocking on the other, something killer in the California area, it was generally seen as though she never gave consent to have that search. It was a good public good, and the European was a public good story. However, there never was consent. So whether I like it and it was good for the public, consent was not given.
Well, now they have the opt-ins.
So now you have the opt-in and saying, I'm fine with that. I do want to know my relatives. I do want to know what's going on. I have nothing to hide. Of course, I don't know what I just unveiled because the technology, what's changing on almost a weekly basis are the applications in the pipelines. We have better ways of analyzing. Now, the resolution of instead of 1,000 reads, there's 10,000 reads is so much better. Much like the space telescopes that had great pictures and now there's a new one that's even better. Why am I using the old one? Because I could still do my work on that. So it is a bit of a mixed bag. I'm watching the Europeans as to where they go with it because they are much more proactive. Privacy is a right. Other questions?
A sequencing machine does it in 4 hours. Not too much detail, but what exactly goes on there? Is it a physical thing, a chemical thing?
So,
Repeat the question.
so the question is the 4-hour sequencer. What's going on? There are chemical approaches. So the picture that I showed was an Illumina instrument. They have about 60% of the market. That is a chemical process where they chop things into bits of 1,000 or so in the reads, amplify it, and this is some of the enzymes that amplify it, and they do what's called a shotgun. They read it all at once, not the little gel stuff. Nanopore is more of physics, and they take the DNA through a reader, somewhat like the movie I showed the second time. And you may have noticed the molecular machine, they said in the movie, first movie, that it runs faster than a jet airplane. So we didn't get into the molecular dynamics of that machinery. They're doing that with a sequencer. And the idea is to get at least a long read as much as Illumina have. And then, so all of these are on microfluidic microfluidic chips. So it's very small sample size. No one in our field has an Erlenmeyer flask of always a purple liquid, and they're not wearing goggles and they're using their hand. Could you at least use gloves? So that is pure fantasy, but sample size is small. Illumina, their instruments cost $700,000, but like Gillette, we'll give you a free razor and we'll bill you on all the supplies. That's where they make their money. Other questions?
Let's do one more question from the audience, and then we'll open it up to everybody to come up and have coffee and mingle. So, one more question?
Okay.
Back in the back. Looks like there's one in the back.
You were drawing a comparison with the banana. I just didn't hear what that was about. You drew a comparison with the banana.
So the banana, we share about 40% of our DNA with a banana. It's the same thing. Now, oddly enough, if you look at a banana and you buy bananas all over the world, there's an enormous overlap because they grew them only one genus and species. So, single point of failure, if there's a virus or whatever on that banana, we get wiped out of all bananas because they've culled it a little too much. We ever did this for plant biology? That is an amazing thing. By the way, for GMO stuff, guys, we've been hybridizing apple trees for eons. That's genetically modified. Go look at what's in the ground versus what's coming out the top. They're two different plants.
So does 40% represent the highest or the closest?
Well, so there's a 40% overlap with the same DNA in a banana as you and I. Now, we have different organs than a banana plant, that's for sure. But there is all the stuff of how to encode, how to unwind the DNA to replicate it, et cetera. It's the same stuff. It's got to be in all of them. So we share a lot of the daily care and feeding of DNA. It's in all of them, including some bacteria. We share that. Okay.
All right, thank you so much. One more round of applause. We invite you to have coffee, mingle, ask more questions. Have a great night, and hopefully we'll see you next month.